This procedure begins with overexpression of the recombinant C-terminus of the purinergic P2X receptor, or P2X2, engineered as a GST fusion protein. Following bacterial lysis, purified protein is incubated with glutathione cephorose 4B beads, and the concentration is read using the Bradford method. Next, proteins from purified rat brain lysate are incubated with the C-terminus of P2X2 receptors bound to glutathione cephorose beads, and protein complexes bound to the C-terminus of P2X2 receptors are isolated via SDS phage gel electrophoresis and excision and are identified by mass spectrometry.
Hi, I'm Harpreet Singh from the laboratory of Baljeet Khak, Department of Physiology at University of California, Los Angeles.
Hi, I'm Sarah Warburton from the laboratory of Tom Vondriska in the Department of Anesthesiology at the University of California, Los Angeles.
Today we'll show you the procedure to isolate and purify the C-terminus of P2X2 receptors. We use these proteins to identify the interactors from the rat brain, and in our laboratory we use these proteins to study the functional consequences on P2X2 receptors.
So let's get started.
The cytosolic domain of the P2X2 receptor consists of the N and C termini. The C-terminus of the P2X2 receptor will be used as bait for the pull-down assay. Also shown is the sequence of the P2X2 receptor C-terminus used for this interaction study. To express the full-length C-terminus of P2X2 receptors, use conventional molecular biology techniques to integrate the coding sequence for the C-terminus of the receptor, residues 353 to 472, into a PGEX4NT1 plasmid. The recombinant plasmid is then transformed into E. coli strain BL21 for expression. The bacteria containing the recombinant plasmid is cultured in LB at 37 degrees Celsius. When the optical density of the culture at 600 nanometers is between 0.6 and 0.8, induce the culture with 1 millimolar IPTG and incubate for 3 hours at 37 degrees Celsius. When the 3-hour incubation is over, remove a 1 milliliter aliquot from the culture and save for later analysis. Spin down the remainder of the culture at 5,000 Gs for 15 minutes at 4 degrees Celsius. After centrifugation, discard the supernatant and resuspend the pellet in 20 milliliters of saline tris EDTA buffer. Transfer the resuspended pellet to a 50 milliliter Falcon tube. The suspension must now be centrifuged at 5,000 Gs for 15 minutes to collect the semi-dry pellet, which is frozen at -70 degrees Celsius overnight. On the following day, resuspend the frozen pellet in 40 milliliters of ice-cold lysis buffer and put on ice for 30 minutes. While the resuspended pellet is sitting on ice, prepare 3 milliliters of glutathione cephorose 4B beads. First, wash the beads with 20 milliliters of PBS and 20 milliliters of TPBS. After that, wash the beads with pure PBS and resuspend them in 1 milliliter PBS. The bacterial suspension in lysis buffer is now freeze-thawed in liquid nitrogen. The frozen sample was incubated on ice to thaw and again purged into liquid nitrogen. This step was repeated 3 times. Next, sonicate the freeze-thawed suspension on ice. After the sonication, incubate the bacterial suspension with a solution containing 4% Triton X100, 10 millimolar magnesium sulfate, and 2 millimolar ATP for 30 minutes on ice. Then, spin down the lysate at 20,000 Gs for 15 minutes at 4 degrees Celsius and save the supernatant. Before discarding the pellet, take a small sample of the pellet with supernatant to analyze the proteins in the cytosol and membranes. Incubate the supernatant with the beads for 1 hour or overnight at 4 degrees Celsius. After their incubation, spin down the beads at 500 Gs for 5 minutes. Discard most of the supernatant, but save a small sample to represent the fraction of unbound proteins. Wash the pelleted beads 5 times with 25 milliliters of TPBS while saving a little of each wash for wash controls. Finally, resuspend the beads in PBS to a 50% weight to volume slurry and store it at 4 degrees Celsius. The protein concentration can be estimated using the Bradford assay as per the manufacturer's instructions and is expected to be 500 micrograms per milliliter. To identify the binding partners of P2X2 CTGST from whole brain lysates, a pull-down assay is performed using CTGST immobilized on the glutathione-coated beads as bait. As a control, use GST alone bound to beads as bait, hereforth referred to as the GST null. Rat brain lysate for the GST fusion protein binding assay was prepared using a standard protocol and lysis buffer, and protein concentration was estimated as 15 milligrams per milliliter by the Bradford assay. Before beginning the assay, pre-clear the thawed brain lysate by incubating it with 30 microliter GST beads for 1 hour at 4 degrees Celsius. After the lysate has been incubated with the GST beads, spin down the beads at 1000 Gs for 10 minutes at 4 degrees Celsius. Save the supernatant and keep a small volume of it as a control. Now, calculate the volume of lysate that should contain 100 micrograms of the pre-cleared solubilized protein and combine this volume of lysate with 10 micrograms of GST-bound beads and 10 micrograms P2X2 CTGST-bound beads. Make up the volume to 200 microliters using lysis buffer. Incubate the lysate with beads overnight in a rotator at 4 degrees Celsius. The next day, spin down the beads at 1000 Gs for 5 minutes and discard the supernatant. Wash the beads 3 times with the 1 milliliter lysis buffer. Then, add modified 50 microliters Lemley buffers to the beads and boil the beads in this buffer for 5 minutes. Spin down the boiled samples at 5000 Gs for 5 minutes and run the supernatant out on a 10% SDS page gel. On the gel, compare the P2X2 CT-associated proteins with those recovered using GST null as bait. The final, critical step of the analysis is to remove the separated proteins from the gel for identification by mass spectrometry. From this point forward, an absence of dust throughout the process is critical to reduce keratin contamination. Begin by cutting the bands from the gel with a clean razor blade. Then, dice the bands into 3 smaller sections and put each band in a labeled microcentrifuge tube. corresponding regions of the gel in the GST null pull-down lane must also be excised in the same manner without regard to band staining to ensure that presence of proteins below the level of staining sensitivity are not missed. Mix the gel pieces with 200 microliters of 50 millimolar ammonium bicarbonate and 50% volume by volume acetonitrile. Then, vortex the tubes for 15 minutes at room temperature. After vortexing, remove the wash solution and repeat. Remove the final wash solution and dehydrate the gel pieces by adding 200 microliters acetonitrile. The gel pieces should shrink and become opaque in color within 2 minutes. Remove the acetonitrile and dry the gel pieces using a speed vac for 10 minutes. The dried gel pieces are next incubated in a freshly prepared 10 millimolar DTT 10 millimolar TSEP solution. The volume of the solution, usually about 30 microliters, must be sufficient to immerse the gel pieces. Allow the gel pieces to be reduced in the solution for 30 minutes in a 56 degrees Celsius heat block. Next, remove the DTT TSEP solution and replace it with a wash solution of 100 microliter 500 millimolar ammonium bicarbonate and 50% acetonitrile. Vortex for 10 minutes. After vortexing, aspirate off the ammonium bicarbonate wash and replace it with 200 microliters of acetonitrile to dehydrate the gel pieces. The gel pieces should shrink and become opaque in color within 2 minutes. Next, to alkylate any free sulfhydryls, replace the acetonitrile with 100 microliters of freshly prepared 100 millimolar iodoacetamide solution. Incubate this mixture for 25 minutes at room temperature in the dark. After the incubation, remove the iodoacetamide solution and wash the gel pieces with 200 microliters of 50 millimolar ammonium bicarbonate and 50% acetonitrile by vortexing for 5 minutes. This wash step is repeated once. Remove the last wash solution and incubate the gel pieces in 200 microliters acetonitrile for 10 minutes. Remove the acetonitrile and dry the samples in the speed vac. At this step, the gel pieces are ready for trypsin digestion. Apply 30 microliters of trypsin solution at a working concentration of approximately 20 nanograms per microliter and incubate on ice for 10 minutes. The gel pieces should become well swollen. Remove the excess trypsin solution and immerse the rehydrated gel particles in 30 microliters of 50 millimolar ammonium bicarbonate. Allow the digestion to proceed for 12 to 16 hours at 37 degrees Celsius. After the long digestion, add 5 microliters of 5% formic acid to deactivate the trypsin and to arrest digestion. Then, vortex the tubes for 15 minutes. After vortexing, centrifuge the tubes briefly to bring the liquid to the bottom of the tube and transfer this solution containing the digested peptides to a clean labeled half-mil tube. Next, cover the gel pieces with about 30 microliters of extraction solution containing 0.1% formic acid and 50% acetonitrile and vortex for 10 to 15 minutes. Add this extraction solution to the new half-mil tube. Repeat this step, transferring the extraction solution to the new half-mil tube. The half-mil tube containing the overnight digestion solution and the 2 extraction solutions is reduced to approximately 30 microliters in the speed vac. This peptide solution is now ready for analysis by LC-MS. Separate the proteins by reverse phase nano-LC on a C18 reverse phase column. Acquire spectra in data-dependent mode with the Orbitrap used for MS scans and LTQ for MS-to-MS scans. Identify proteins by searching the data against the RAT IPI database using the Sequest algorithm integrated into the BioWorks software package, and then manually inspect the spectra. Only proteins that are present in each of the 4 sample replicates and are absent during GST null pulldowns should be considered for further analyses. Here is the Cipro stain gel showing a spectrum of putative proteins that interact with the C-terminus of P2X2 receptors fused with GST, indicated by the blue box. Control lanes for GST alone, indicated by the yellow box, and glutathione cephalospeeds alone are also shown. The arrows indicate examples of unique bands that were excised for further analysis by mass spectrometry.
We have just shown you how to express and purify the C-terminus of P2X2 receptors. We have also shown you that how to perform the pull-down assays and identify the protein interactors.
It's important to remember when doing this procedure that you wear gloves and a hairnet to reduce the keratin contamination in your samples.
So that's it. Thanks for watching and good luck for your experiments.